As the result of speciation processes operating over billions of years, we find ourselves in a world in which the information content of life, represented by the DNA of all living things, has become diversified and organized into millions and millions of different packages, millions of different species, each carrying its own unique variant of that information and each having its own unique form and place on the planet. From my point of view, the diversity of life ultimately is what makes biology both fascinating and a little daunting. It's fascinating because in spite of the commonalities shared by all living things, commonalities we've emphasized throughout this course, each species is at least a little different in the ways that it goes about its business of being alive. And more often than not, it's simply astounding how many radically different ways there are to be a living organism. But this wonderful diversity also can be a little daunting. How can we keep track of all of those different kinds of organisms and what they do to make a living in this world as we try to understand more about biology? The only way we can hope to do this is to somehow classify living things, much like a librarian keeps track of the books in her library, by having some consistent and relevant system for cataloging the collection. Now, there are many relevant ways in which we can organize books in a library, by subject, for example, or by author, or by any other number of criteria that we found useful for retrieving particular entries. But how can we sort out the enormous diversity of life on Earth? That is, how can we catalog this diversity in a relevant fashion? In biology, the most relevant and most useful way to classify living things is according to their evolutionary history. In today's lecture, we're going to take a look at how we can learn something about the evolutionary history of groups of organisms, both as a way to classify them and as a way to gain a different kind of insight into the concepts of information and evolution, which have served as the theme for this first third of our course. Classifying organisms according to type is undoubtedly the oldest activity that we would consider as some sort of biological science. The Greek philosopher Aristotle, for example, went to considerable trouble to classify all living things according to, in his case, a scale of increasing complexity, or what we refer to as a scala naturae. In Darwin's day, taxonomy, the naming of organisms and their classification relative to each other, was a dominant field in biology, and in fact, this occupied much of Darwin's own professional life. Today, the area of biology that concerns itself with the classification of organisms in an evolutionary context is called systematics. Let's begin our discussion of systematics first by thinking about how we might classify organisms, going back to that example of the two species of sparrows we've talked about, song sparrows and chipping sparrows. I talked about these a couple of lectures ago to introduce the problem of defining species. Now, at that time, I made the point that you would have no trouble classifying these as two separate species based on their obvious differences. Now imagine I give you a slightly different task. Let's say I gave you these two species of sparrow, and I also gave you a duck and a vulture. And I asked you to sort these organisms out relative to each other. I'm sure you would have no trouble in this case pointing out that the song sparrow and the chipping sparrow are more like each other than they are to either the duck or the vulture. Now let me add a few more organisms into the mix. Let me give you, to your growing collection of organisms, a dog, a cat, and a lion. Now I want you to tell me how to classify these organisms, and the way you would start arranging them gets a little more complicated. You certainly would still want to place the sparrows in one group, but you'd probably also want to place all of the birds, that is including the duck and the vulture, in a more inclusive group relative to the other animals that I've added. This is because, in spite of their differences, the birds are all more similar to each other than they are, say, to the dog or the cat or the lion. You'd probably also have sorted the dog and the cat and the lion relative to each other, putting the cat and the lion into a group that's a little bit tighter than, say, relative to the dog. Now let me add just one more organism to your list. In this case, I'm going to add a fruit fly. And when I add this fruit fly and ask you to tell me how these organisms might be sorted out, you would be tempted probably to somehow want to group together all of the birds and the cat and the dog and the lion, the carnivores, in some even more inclusive group relative to the fruit fly, because these organisms all obviously, as different as they are, all obviously share a few things in common. For example, they all share internal skeletons. They all share backbones, things that the fruit fly doesn't have. Now, we could go on with this exercise for a long time, but I think we've done enough to have 2 points emerge. The first point is that we tend to classify things relative to each other on the basis of features they share in common. And this isn't at all surprising. For example, we would put all of the animals that had fur and hair together and call them mammals, and this would give us a group that would include the cat and the dog and the lion. My second point is that our classification is hierarchical. The sparrows are more similar to each other if we compare them to other kinds of birds, but if we add dogs and cats and lions, then we want to somehow make an even more inclusive group that includes all the birds relative to these other species we've added. We've created a hierarchy of classification, basically a nesting of groups within groups. Now, we usually classify organisms according to a hierarchy, with the lowest level of the hierarchy being organisms that are very similar to each other, and progressively higher levels of the hierarchy grouping together organisms that are progressively less similar, but still more similar to each other than they are relative to other groups of organisms. Now, actually, the scientist who kind of established the way we think about this was a Swedish botanist named Carl von Linné, who is usually referred to by his Latinized name, Carolus Linnaeus. Linnaeus devised the hierarchical scheme that we use today to classify organisms. And this hierarchical scheme was based on placing the most similar organisms together in a small unit and then placing these units themselves into ever more inclusive, larger units. The smallest unit in Linnaeus' scheme is what we call a species. Species were grouped into genera, that's genus if it's not plural, genera into families, families into orders, and with increasingly more inclusive higher levels being referred to as classes and phyla and kingdom, so forth. Now, we don't need to worry about remembering those names for our purposes, but the point I want to make is that in the Linnaean system, each organism received a series of names from which we could not only know its individual identity as a species, but also know the nested groups to which it belonged based on similarities among those groups. For example, let's go back to the song sparrow. It's called Melospiza melodia, with melodia being the species name within the genus Melospiza. And when we talk about the scientific names of an organism, we usually refer to its genus and species, Melospiza melodia. The genus Melospiza is a member of the bird family Fringillidae, which is in turn a member of the bird order Passeriformes, which is a member of the class of all birds put together, which we call Aves, and so forth up on the line. We still use Linnaeus' system for naming organisms and groups of organisms, but unlike Linnaeus, we're no longer simply interested in developing a catalog of species for their own sake. Ultimately, what modern evolutionary systematics seeks to uncover has less to do with how similar two or more species are in and of themselves for their own sake, but more to do with how related those species are in an evolutionary sense. Similarity comes from being related, as Darwin's view of common descent would suggest, but we're interested in the evolutionary history, not similarity for its own sake. We refer to the evolutionary history of a group of species or a group of higher order taxa. Taxa is just a word we use to label any level of hierarchy in this classification scheme. We refer to the evolutionary history of a group as its phylogeny. And what we want to get to by the end of today's lecture is to give you a little bit of an idea about how we can know evolutionary history to develop a phylogeny. Well, classical approaches to taxonomy and systematics assumed that similarities between species or higher taxa must reflect a shared evolutionary relationship. Or if they didn't assume it, they didn't really care so much. They just said if it's similar, we'll use it. If it's a characteristic that's shared, looks similar, let's use it to classify organisms. But there are two reasons why this doesn't work for us. The first one was figured out long ago. It was figured out in Darwin's day. And that is, that, or even before, frankly, not all similarities among species are the result of those species being descended from a common ancestor. Some similarities do come from common descent, but not all. Instead, similarities that we observe between organisms may equally well arise from a common set of selective pressures acting on otherwise unrelated organisms, leading to what we call convergent evolution of similar adaptations. For example, if you compare the flippers of dolphins with the fins of fish, you find that these structures actually resemble each other, at least superficially. But we know that flippers and fins are derived from completely different structures. The similarity between them arises from their similar function and thus from the fact that natural selection leads to similar kinds of adaptation in these two very different structures. Structures that are similar because of convergent evolution are referred to as analogous structures. Structures or other features of organisms, anything that they share in common, that are similar due to common ancestry, we refer to as being homologous. Now, we've already talked about homologous structures earlier as one of the several kinds of evidence 19th century comparative anatomists offered in support of the idea that species evolve in the first place. The example I used was of the forelimb bones of different kinds of mammals, all of which share similarities reflecting not some common selective pressures, but instead common evolutionary history. For example, the forelimb bones of humans, cats, whales, and bats look quite different because those forelimbs are used in different ways, but in spite of the differences, they nonetheless share similarities in terms of their position, aspects of their shape. It's just that they've been stretched and twisted in different way by different selective pressures. If we recognize homologous structures, homologies like these, we can potentially use those as useful information to help determine the evolutionary relationships among a group of organisms, because those similarities do reflect common descent. Now, determining whether structures are homologous or not actually itself is quite a bit of work. Biologists, when they make the statement that two homologous structures are homologous, typically have to rely on a variety of kinds of evidence, not just structural similarity or appearance, but also evidence coming, for example, from developmental patterns or aspects of how those structures are related to other structures that are in the same area. Or increasingly nowadays, we use genetic evidence to determine whether two structures actually are related through common descent or just look similar. And if they are related, we refer to them as homologous. Now, I said a moment ago that there are two problems with using similarity for determining evolutionary relationships. The first was this problem of determining which structures are truly homologous. The second problem is this: even homologous features are not always useful for helping us to understand the evolutionary relationships among a group of organisms. Well, consider a simple example. Let's say I'm interested in the evolutionary relatedness of the three carnivores that we talked about earlier: cats and lions and dogs. All of these species share many similarities in common that are homologies. For example, fur, or the ability to control their body temperature, or providing milk for their young. These similarities are homologies, but they're not useful to us in terms of figuring out the relative evolutionary relationships among those three organisms, because they all share them. It doesn't sort anything out. On the other hand, if we looked, for example, at the presence of retractable claws, a similarity that is homologous across all of these species because the claw structures are homologous, but which differs clearly in the cat and the lion because they can retract their claws from the dog, which can't, then we can use that kind of homologous structure that differs among some of the members of the group we're interested in and is the same among others to determine something about evolutionary history. We would, in that simple example, in that simple case, for example, suggest that that tells us that the cat and the lion are somehow more closely related in their evolutionary history than the dog, and we would be right about that. Well, Let's ask how we can use these kinds of shared homologous similarities in a more formal context to understand something about the evolutionary relationships among a group of organisms, or to put it another way, to construct a phylogeny for a group of organisms. The key insight into this problem was developed in the mid-1960s by a German entomologist named Willy Hennig. Willy Hennig was the person who almost single-handedly established a field we call today phylogenetic systematics, which basically means the classification of organisms based on their phylogenies. Phylogenetic systematics is an approach to classifying organisms that is explicitly and exclusively based on inferences about their evolutionary history, and to make those inferences is looking at how they share similarities and differences among homologous structures. Now, Hennig realized a few things which are actually pretty obvious to us, I think, today anyway, of where we are. Hennig realized, for example, that the evolutionary history of a group of organisms would be reflected in changes of structures or other features that are observable characteristics of those organisms. This is nothing new to us. Now, following Hennig's convention, the one he introduced in phylogenetic systematics, we're going to refer to structures or other features that we're interested in using in this way as characters. So we will be talking about characters, which are features that organisms share or don't, and we'll be talking also about character states, which are different ways that those characters might appear. So what we're really looking at here are homologous similarities that provide us with characters to look at relationships among organisms. In the 1960s, the kind of characters that Hennig had available and with which he developed this idea were largely morphological characters, things like the shape of bones or the size of wing parts for an insect and so forth. But characters can be anything that we can measure about an organism, and increasingly characters now in modern phylogenetic systematics are DNA sequences, because we can use similarities and differences among DNA sequences that we know to be homologous across organisms equally well to look at their relationship. Now, Hennig argued that through the course of evolution, some characters will change in some species while others will not, and that those characters that change or not will differ among different species. As species in a group change over time, there will be different patterns of the degree to which characters that those species share do or don't change. And he suggested that we could use the differing patterns of change of commonly shared characters as a way to infer something about the phylogenetic relationship of a group of species. Now, we refer to characters that have changed over time as derived characters. Characters that have become different than what we presume they were in some earlier or ancestral species. Those characters that have not changed in an organism or a group of organisms, we will call usually ancestral, but often we say that these are the primitive character states. Now, I want to point out that primitive is often used in this context, but it's in a sense a poor choice of word, because when we think about primitive, we're often thinking that this must mean somehow less well adaptive, not as good, or something like that. All primitive means in this context is the more ancestral version of a trait, or what we're calling a character. So each species is going to have some characters that have changed a lot and some characters that have changed a little. Those characters that have changed a lot within a group of organisms we call derived character states, or those states of characters, I should say. So each species in a group is going to represent some mixture of primitive characters, which it shares with its ancestor species, and derived characters, which it does no longer share with its ancestor species. Now, none of this should seem very earth-shattering. In fact, it's just what we would expect from our discussion of evolution so far. But what Hennig did was suggest we can use a formal analysis of formally defined characters and character states to actually derive some evolutionary relationships among a group of organisms. And his insight to do this was relatively simple. He suggested that the characters that are particularly useful for determining true evolutionary relationships among organisms are those derived characters that are shared to differing degrees among organisms in a group, the so-called shared derived characters. So we want to find characters that we can work with. These we need to assume are homologous across the group. We want to find characters that have changed, have become derived in some members of this group and not others. And then we want to look at the patterns to which members in this group that we're interested in analyzing do or don't share these derived characters. Now, Henig's method for doing this is actually a statistical method that, since he originally developed it, has been elaborated quite a bit. We could work an example by hand, although I don't intend to do that right now. But in practice, the way systematists do this is largely with very complex computer algorithms, because they'll use numbers of characters, huge amounts of data, especially if they're using genetic data, and they will be including many, many kinds of organisms in their analyses. So what the common practice of phylogenetic systematics is to use a number of computer algorithms that have been developed to actually essentially give you a statistical probability of what the best fit of characters to different possible phylogenies are, and use those phylogenies and the statistical estimation of their accuracy as hypotheses for the evolutionary relationships among the group. Now, we could work a simple example with just a few characters and actually come up and cook the numbers in such a way that we came up with just one single way that those individuals in a group were related based on those characters. I don't want to teach you how to generate a phylogeny, but I want to give you a sense of what's involved in the process. So let me give you an idea of the kinds of information we need to work with. I'm going to use a relatively simple example. I'm going to ask us to consider what the evolutionary relationships are among 7 species. We'll talk about, for example, a lamprey, which is a jawless fish, which we can assume is a relatively ancestral member of this group that we're talking about. A trout, which is a more derived fish, a so-called bony fish. A lungfish that actually is quite different, that, as its name implies, uses lungs in addition to gills to obtain oxygen. A turtle, a cat, you're familiar with these. Then we're going to throw a gorilla into the mix, and a human. Now, we've got a group of organisms, and we want to ask the question, how can we tell which are more closely related to which? How will they fit on some tree that we develop, some phylogeny? And to do that, we need to pick some characters that we know are homologous across those species, but we also know differ in differing ways. So we could choose characters like, for example, the presence or absence of jaws. In this case, the lamprey, which is so-called a jawless fish, lacks a true jaw, and all of the other species I named have jaws. So that's an example of a character that is shared by all species, but not one of them, the lamprey. We could use limbs, the presence of limbs as a character. We could use the presence or absence of hair. We could use the presence or absence of a lung, or of a tail, or of a shell. These are very simple characters, and it would be no trouble to measure them or to actually just make note of which species had those characters or not. The point of just choosing some characters like that is to illustrate how these characters, which are homologous across all members of the species, may or may not actually be shared by them in the way they appear. So, for example, cats and gorillas and humans all have hair, while the other 4 species don't. Another example would be to look at the presence of a tail. The lamprey, which we're assuming is a relatively primitive species, has a tail, as does a turtle and a cat. A lungfish and a trout, we would say, have tails based on their anatomy, but in fact the gorilla and the human don't have a tail. And we could keep track of these species and these characters by making a table, for example, where we put the species that we're interested in as labels on the rows of the table, and the characters that we're interested in as labels on the columns of the table, and then go ahead and just fill in the table, designating whether those species had or did not have those particular characters. Now, there's a couple of assumptions we've made here. I've already mentioned one, but I want to repeat it, and that is the first one, that these characters we are assuming are homologous. To do this analysis right, we need to know that they're homologous. We need to know that hair is a homologous structure between cats and gorillas and humans. And, of course, we know that's the case for all of these simple characters. The other assumption we're making is to designate one of the species in our mix as being the most ancestral species. I've already mentioned that we're assuming that the lamprey, in this case, is an ancestral species. the primitive species. Again, not designating primitive as somehow necessarily less adaptive, but simply saying that we're assuming that it is more ancestral than any other species in the mix. Now, we will call the most primitive species an outgroup. We call it an outgroup because we're sort of keeping it outside the analysis. It anchors the rest of the analysis. The value of using an outgroup species to anchor an analysis in this way is that it allows us to designate which of the characters in the analysis are derived and which aren't. Because we can look at the character states for our most primitive species, our outgroup, and by definition assign those states as being the primitive states. And then all other states we will consider to be derived relative to them. So for example, lamprey lack hair. So we would assume, or actually define, the lack of hair as being the primitive state. And therefore, cats and gorillas and humans, which have hair, share a derived state. Now, it doesn't need to be that the absence of a trait is somehow primitive. In this case, actually, lampreys have tails. So we would, in this case, have defined the presence of a tail as being the primitive trait. And then, therefore, the absence of the tail, such as this characteristic of gorillas and humans, becomes the derived version of the trait. Now, once we've got our derived traits worked out, then we ask, how do the different members in this analysis, the different species in the analysis, share or not share traits? It's the shared derived traits, the degree to which organisms share different derived traits in specific, that we use to determine phylogenetic relationships. For example, humans and gorillas share 5 of the derived traits in our analysis. They share the traits of having jaws, limbs, hair, lungs, and not having a tail. All of those are derived traits. On the other hand, humans and cats only share 4 derived traits. Cats do have a tail, the primitive traits, while humans do not. This might suggest, correctly in essence, that humans are evolutionarily closer to gorillas than they are to cats. And if that conclusion seems simple, it is. It's the beauty of Henig's method that it bases inferences about evolutionary relationships on simple patterns of how different species share these derived characters. Now, as I said, to actually go from there to generate a phylogenetic hypothesis would take quite a bit of analysis, and we're not going to do this. But instead, let's just consider what we might learn if we actually did that exercise. What we would get out of this analysis is something that would look a little bit like a tree. It would have a branching pattern that goes to some base at the bottom of the tree, and then each branch, as it reaches up, would end with the name of one of the species in our analysis. That's not too surprising. I mean, people have been drawing evolutionary trees of life, or just trees of life, for actually centuries now. The difference here is that the specific branching pattern we observed is derived from the data we've put in relative to the numbers of characteristics, or characters I should say, that the species in the analysis share. It's not an arbitrary branching pattern, nor is it just our own guess as to what's related or what isn't related. It is an estimate of what's related or not based specifically on data that we've put into the analysis. Some of the outcome of this would be obvious. We would learn, for example, that humans and gorillas are more closely related to each other than they are, say, to cats. That's pretty obvious. But we would also learn from this kind of analysis some sorts of unobvious things. For example, if we looked at this analysis in detail, we would be able to infer that lungfish are more closely related to gorillas and humans than they are to trout. That seems surprising. They're both fish. The way we know that, and the essence of how we understand evolutionary relationships, is to look down into the branching pattern of that tree and ask, where are the nodes, where are the branch points that join different species? The branch point that joins, if you were doing this analysis, lungfish on the one hand with humans or even gorillas on the other, occurs at a higher point in the tree than the branch point that we would have to go to find a connection between lungfish and trout. And it's in this way that we actually can know something in detail about the evolutionary relationships of organisms in this group. Well, the advent of these kinds of methods really transformed the field of taxonomy, and it turned it into more of an evolutionary science than a classification science. And the methods that have been derived are now very primary in the way that we study evolution, because we can use the patterns of relatedness that we see from phylogenetic analyses like this to infer things about how evolution must have occurred long ago in the past with species that no longer exist. I think it's interesting that the shared derived characters that we use in this way actually represent, again, biological information, and in this case, on the highest level of the hierarchy of biological organization. The information responsible here still ultimately resides in the DNA, but the patterns we're interested in now even transcend single species, but instead are patterns that will unite entire groups of species in different ways and tell us how we can draw lines between species, among species, as we determine how various organisms, how the great, enormous diversity of life on the planet should be arranged as we try to understand it in biology.